About micro, she didn't like micro.
She had a horrible time at micro. I said, I'm having a great time. She's like, I don't understand how.
I feel like micro is truly something that you have to like, it is what you put into it. You have to show up and do your homework and assignments and all that stuff.
Yeah, people don't like this class. I'm not gonna lie. I'm aware of why people don't like this class. They don't like it because they have to work at it. Yeah.
It makes me feel better. I hate anatomy so much and I don't hate this class, so.
Honestly, I think this class is like the least amount of work that I've done on all my classes, but it's just more studying. That's all it is.
Yeah.
When it comes to homework and stuff like physiology, you know, we have like 7.
You have to put in the time to get the assignment done, and it's not.
And
Also,
yeah.
and it,
do 1 5-hour.
yeah, and it makes you feel any better. This girl just hated life and she just complained the whole time. So I think it was just her personality.
Yeah, if it makes you feel better, I don't know who it could be because I think there's so many people that it could be.
That's my cousin who took you said she had a great time in here.
Yeah, it just
And I'm
depends.
singing your praises all throughout everywhere where I'm like, where I work. So I work at some Red House,
Uh-huh.
and there's a bunch of kids there that are like thinking about college and thinking about going. And I'm like, if you go to OSUPIC, do not take physiology, but take micro.
Yeah.
Physiology stuff.
It just depends. I really think you have to be suited to it. Like, just some people aren't built for the work. So it's what it is. I don't know.
Oh, so about my professor that I emailed you about,
Mm-hmm.
the guy, the president over there at
Yeah.
Stillwater has no dictation over OSU.
That's the only info that I could find.
Yeah, I emailed him, he goes, I don't have it.
Our last leaders, all of us. I have anxiety section. I just
Appointment over there, over at OSU.
Well, yeah, I mean, I don't know that department well enough for me to even like give you any other information other than, yeah. Did you try emailing Brian, whatever? I don't know. Oh, it is. I have no idea who else to contact.
He said that they've been calls like this before, and then he'd get back to me.
Yeah.
So nothing great is.
Yeah.
And I talked to her.
I have, so when we were talking about this, of course, I have, we're, busy with all this stuff now. Now, all I have to worry about really is reviews and writing the exam, and which that's not gonna take me all that long, right? So I have time now to figure that out. So I'll look more into it and see if I can get, but if still you don't have anything graded, like if so, you're just gonna be surprised, like,
And like my final lab project is supposed to go on. So what's that, first project?
And it's not even graded.
My first one's like, and my second one was due yesterday.
Okay, I'll talk to, well, I know who to talk to. I'll talk to Dr. Rhodes in my department because he'll know who the department head is for that, but.
I've emailed her, gone to office hours and.
What's her name?
They open.
Just so I know who to,
I think.
I'm not trying to get her in trouble or anything, at least like, though, I mean, she should be in trouble, but like, I'm not trying to go that way.
Is it LP?
Tamara Williams.
Okay.
Professor, yes.
Mhm.
I got my nursing letter.
Oh, yeah? Good job.
Yeah.
I always tell people, like, if you're making it into here and you think you're doing okay, and then you get the letter, it's pretty good news. But I have a lot of students that come to me like, they'll be like, okay, well, I got accepted to nursing school, and this is the, I just have to get this, I'm in nursing school, and I just have to take care of this class, and I'm like, do you know what you're getting into? It's not what you think.
Yeah, I was like,
Yeah.
my cousin's in her second year right now and texted me about it. She did so well in this class, and she's like, Rachel, I need you to study until your eyes bleed. She's like, it's kicking my ass. I was like, cool, that's encouraging, I got it.
Yeah,
Pharmacology is covered.
I do try to warn people of that. It's like, you think this class is hard, it doesn't get easier after this.
I know, it gets so hard.
Yeah. So.
I'm just gonna cry, and I like to be crying.
Anyway, so do you guys, okay, since it's just you guys here, and I figured I wasn't gonna have a full class, but do you guys want to play like against each other like we usually do? Or do you want to try to do something cooperative? Or do you want to try teams? Anybody care about any?
Michaela's almost here. She's coming from Parkgate, and she's leaving.
Okay, we'll just do the regular way then.
Alex, are you guilty, my dude?
No, I just hate sitting forever.
Yeah, I know what you mean, man.
My monitor does not all work.
That's what I don't wanna do.
There's 8 of us. I'll wait till we get in and then we'll get started. All right. Pretty simple review. I try to have 6 questions from each chapter, so we have a good rundown, remind ourselves what we learned. All right, so remember units 1 and 2, mostly these are covering what microbes are and how they function. So unit 1, we're going to go through chapters 1 through 5 and then 7. But intro to micro, tools of micro, chemistry, and then bacteria, eukaryotes, and viruses. So really just the basic intro stuff. All right, microbiology is the study of. All right, great job. So this is going to be things that are too small to be seen with the naked eye. We need something to magnify them in order to look at them. So yeah, microbiology study of that. I would say for cells, that'd just be cell biology. Literally, they have their own kind of area. But yeah, when I smell cut grass, it reminds me of like early 2000s. There was this perfume that the Gap came out with that was like literally cut grass. Yeah.
Yes, I know what you're talking about.
All right, all disease-causing agents are made of cells. All right, so not true. This would be false, and most of you guys got that. And that's because why? Because viruses are not cells, right? They're just protein-coated nucleic acid, and then prions also, right? That's technically disease-causing. They're just protein. All right, what is the correct term for disease-causing microorganisms? There's a term for the disease-causing microorganisms or agents. Right, so that term would be pathogen. Great job. We're getting places now. All right, if you're sick with a communicable disease, what's the one thing you know for sure? Yeah, right. Out of these choices, what's the one thing? This one is, I think, through your choices. Alrighty. So this one, if you're sick and it's communicable, we know that it's caused by some sort of germ, right? It could be bacteria, but it could be a virus, but it could be protozoa, you know, it could be fungi, all of that. So we know that, and there's a term for this, that's germ theory, right? That germs cause communicable diseases. I know, it's been a long time.
I'm so sorry, that was loud.
The correct way to refer to the cause of the plague is Yersinia pestis. What system dictates how to write this?
Oh, shit.
I know, right? This is the stuff we never come back to because it doesn't matter to me in my mind a lot of the time, but we have to come back to it.
My sister-in-law has that mask under her bed.
Oh, yeah.
I thought it was for weird doctor foreplay for her or other, but it's not. I thought that they're both doctors. Why do you hide that under your bed?
All righty. So this one is binomial nomenclature. You guys did great with that. All right, true or false? In taxonomy, kingdom is larger or more general than domain.
I have this song set in my head all day yesterday.
Yeah. So this one's going to be false. The reason it's false is because, well, domain obviously is the biggest, but domain includes archaea, bacteria, and eukaryotes. So that's the big picture thing. And then kingdom kind of applies to the eukaryotes. So kingdom, phylum, class, order, family, genus, species. That's how we would put under kingdom, right? But domain is above that. All right, inoculate, incubate, inspect, identity. identify is what it's supposed to say. What is the missing I? So which one are we not having? Yeah, good job. That's going to be isolate.
Big brain, they don't.
All right, what type of media only allows certain kinds of microbes to grow and inhibits the growth of others? What type of media would that be?
Just made it weird.
Yeah.
Yeah.
I didn't do this, just to be clear.
Mentality.
All right, this is correct. It's going to be selective. I'm glad we carried something over. And I'm glad some of the information from the lab kind of carried a little bit. There's something, but the ocular lens of the microscope generates the real image. Is that true or false? This one was on the lab exam, right? We had that.
Not wrong.
Yeah.
I'm not wrong.
Yeah, so the ocular lens of the microscope generates the virtual image. All right, if the objective lens is 40x and the ocular lens is 10, what's the total magnification? Believe it or not, we did actually learn this in the chapter stuff too. I'm not just, some of this will be at least easy though. You'll follow it pretty well. All righty, good job. What is the purpose of foil immersion? All right, good job. That's going to be reducing light scatter because it more closely mimics the glass itself. So the light rays don't scatter when they're changing between those. Right, a negative stain stains the background because it's repelled by the cell surface. Is that true or false? For the aspects of it, do negative stains even stain the background? And if they do, is it because they're repelled by cell surfaces? All right, so this one's true. That's why negative stains do the way they do. Remember, the outer surface of cells are negative from those phospholipids, right? The phospho part is negatively charged. So negatively charged stain is repelled by that and will stain the background as a result. All right, what type of chemical bond involves transferring an electron from one atom to the other? Complete transfer. All right, so this one is ionic. In ionic bonds, we'll have that full transfer. And just think of how we have the sodium and the chloride atoms, right? The sodium being positive because it gave up that electron to the chloride, which now has the negative because it has that electron because of that transfer of that electron. Now, the covalent bonds, they share the electrons. So be sure, that's the term that I would be aware of when we're talking about ionic versus covalent. If it says sharing of the electrons, that's going to be the covalent. Okay. Great. Carbon dioxide is an organic molecule. True or false? CO2. Anybody watching Fallout?
Yes,
Yeah.
it's so good.
It is really good. Yeah. All righty. So carbon dioxide, it is not organic. It is inorganic. Why? Because we don't have a carbon hydrogen backbone really going on. It's just a carbon and two oxygens. Just having one carbon isn't enough to say something is organic. We need that carbon hydrogen backbone. Okay. Olive oil is a great example of an everyday hydrophilic molecule. Is that true or false?
Oh.
All right, so oil, they say mixing like oil and water because they don't mix because oil is hydrophobic. It was afraid of water, right? So olive oil is hydrophobic and hydrophilic. That's why that's false. Some of you guys caught that little term switcheroo. All right, which pH is the most acidic of these choices? Right, most of you guys got this one. I'm glad for it, since these are not traditional acidic pHs. You still got it, so it's gonna be that 7. It's the most acidic of them all. Which pH would have the highest hydrogen concentration? All right, so higher hydrogen concentration, the more hydrogen ions you have, the more acidic you are. So that's going to be 2, pH of 2. Because it is I could show you the math equation, but I'm not going to get into it because I'm not going to make you use it, so don't worry about it. Which level of protein structure features alpha helices and beta sheets?
Oh, I hate this.
All right, so it's going to be secondary, right? So we'll go through just real quick the concepts of these. Primary is going to be the amino acid sequence. Secondary are those alpha helices, beta sheets, those initial folds that we're going through. Tertiary is going to be those overall bigger folds of how those things from the secondary are going to interact with one another. And quaternary is when you have more than one polypeptide coming together to form a functional unit. All right, true or false? Bacteria and archaea are always single cell. All right. So this one's true. So they're always single cellular organism. Even if they make a biofilm or something like that, they're still technically single cellular organisms that can function on their own without the other. It's not like you take a liver cell and you take it out of the liver. Can it go and live and be its own cell forever after that? No, right? It needs the rest of the body. So that's what we're talking about with multicellular versus single
Really
cell.
fun too.
All right, what cellular arrangement is featured below? Yep, strepto. Good job. I'm very proud of you guys. Those chains gave it away. So which flagellar arrangement is shown in this image? So if you guys thought that you never have to think about it. All right. It's amphitrachus because it had a flagella at each end. And remember, if we have at each end, that both word is always going to be amphi. So that's how we remember it. All righty. True or false? Gram-positive bacteria have a thick outer membrane. Read the term. Read the words up there for y'all choosing your choices. Something I was gonna say. I don't remember now. Alrighty, so this one's false. They don't have a thick outer membrane. They have a thick cell wall, peptidoglycan cell wall. Be sure you're reading your choices. That's the sort of crap that gets people on the test too.
As soon as you started warning, I was like,
All bacteria are capable of making endospores if nutrients are scarce. Is that true or false? What do we know about endospores? And what kind of bacteria make them? All right, so this one is false. I'm glad that most of you guys got this one. It's going to be Bacillus and Clostridium that do this.
such a strong word.
Yeah, it is. Yeah, so I think it's actually kind of smart. So which group would Neisseria meningitidis, Graham Stain pictured, fall into?
We learned this year before.
We surely did. We surely did. I'll tell you how here in a moment. But this is Neisseria meningitidis Graham stain. It's red, right? Pink, but red. So at first I wasn't even going to give you like that. It was gram negative. I was just going to ask for the question. That would be a dick move, but it is. It is technically in, you know, the notes or whatever, but Neisseria meningitis, it causes meningitis, right? It causes the one with the rash, it's a really, really bad one, it's deadly. Yeah, gram negative, Diplococcus. That was this gram stain, gram negative, right? So here's these groups that we had to choose from: Tenericutes, Firmicutes, Gracilicutes, and Mendozacutes. And remember how I break it down? Mendozacutes, Joey Mendoza was cute. That was a long time ago, so
Oh,
that's
yeah.
archaic. Yep, this is that, back again, right? Archaea. So the Firmicutes, they're firm because they've got that thick cell wall. That's gram positive. Then the next G for gram is going to be the gram negatives. That's Gracilicutes. And then Tenericutes are the ones without the cell wall. So things like mycoplasma.
Oh, cow.
I know, it comes back. You remember it now. It's just
Dust off my brain.
been a while, yeah. So hopefully when you guys get to studying that stuff, it'll come back. Some eukaryotes can use cilia for locomotion or motility. Is that true or false? Can eukaryotes use cilia for locomotion? All right, so you guys did pretty good. They can. But we remember about bacteria, right? They can only use flagella,
Okay.
right? But the eukaryotes, they can flagella, they can cilia, they have pseudopods, and then they have groups that don't move. So those are the options for them. All right, what is the function of mitochondria?
I have no idea. I don't know why we remember that. In deep
Good job, guys.
cell, yeah, literally.
Only thing you're taught in like high school.
Yeah, I know, it's one of those that always carried for me too. So which step of mitosis is pictured? Sorry, I apologize because I know most of you guys are not gonna get, I say most, I don't know if anybody will. It'll be luck if you do, fellas. Mitosis. We remember PMAT, right? Mhm.
Yeah, I remember seeing that.
The puppy pees on the mat, but what is going on here?
Oh, I was.
Really genuinely impressed with you guys here. That is in fact anaphase. So remember prophase, we're just getting everything ready. We're winding up the chromatin and stuff. In mat, so the next one is M, metaphase, we line up the chromosomes. In anaphase, we pull them apart, and in telophase, we start moving into making this new cells after it. So, yep, that's it. So good, not bad. Genuinely surprised.
That's so sad.
What is the term for the overall structure of an entire fungal mass?
Dang.
What is it, Resident Evil? I feel like that's. I don't know if any of you play Resident Evil, but this makes me think of it every time. So this is okay. It's mycelium. So this is when we're talking about structure of fungi, the hyphae are like the little hair-like growths off of them. The overall structure of it is called the mycelium. The sporangiophore and conidiophore are just for asexual reproduction. That's where they make their spores from. One is covered and one is open. The open one is conidiophores.
You said the hyphae was just.
It's the hair-like growths, like, you know, whenever you've seen, like mold, like if you look at a growth of mold and it looks hairy, those individual hairy like appendages that it gets, those are hyphae. Yeah. What is the term for the free living metabolically active form of the protozoa? All right, most of us got this one. This is trophozoite. Remember, trop means to eat. So yeah, the eating version of these guys. We know it's not endospore vegetative. That has to do with the bacteria, or at least we should know that it has to do with bacteria. So it couldn't have been those. So we know it had to be trophozoite or cyst, and trophozoite is an eating form. Cool. True or false? Helminths are animals. Are they?
We're Steph on it.
I don't know, we're both. No.
So these are all very interesting conversations. So these guys, helminths, they fall into the kingdom Animalia, just like bugs. Yeah, insects fall into Animalia. The Animalia doesn't necessarily mean mammals, right? Mammals, Mammalia falls under Animalia there. So yeah. Anyways, that's how you can think of that. Not that it ever matters. You're probably never going to revisit this after this class, but. All right, quiz. There could be more than one answer to this one. Select the types of genomes that viruses have been found to have.
Oh.
This one should be easy given the choices, but we're not going to judge. All right, you guys did great. You guys did great. It is in fact all of them. Usually when I do these all of them ones, people do terribly on them, but you guys did good. No fooling you. All right, true or false? All viruses have capsids. All right, so this one's true. I can actually tell you, for those of you who are choosing false, probably what you were thinking of. Either you just didn't know and you guessed, or you were thinking probably like, yeah, they don't all have capsids, right? At the outside, yes, that's probably what you're thinking in your mind. They don't all have envelopes.
Oh.
Right, so remind ourselves of that one. The envelope is that lipid bilayer coating. The capsid is that protein coating over the nucleic acid, whatever that nucleic acid is. And we learned just from the other slide that could be any. So yeah, good job though. Good thinking. You had the right ideology
I
there.
had two answers.
Where does the viral envelope come from? That lipid bilayer that you were thinking about, called the envelope, where does it come from? All right, the virus steals it from the host when it buds. So it just covers itself with it like a nice little cozy blanket as it's pushing through the outside. All right, the lytic phase is the phase where bacteriophages multiply until they burst out of the host cell. Is that the lytic phase? Is that what it's called? Basically, is the question. I don't know where this GIF came from, but it's fantastic. Yeah. Yeah, it was in the Kahoot thing. I just like looked for virus or something, and then this is, I loved it. It was perfect. Yes, so the lytic phase is in fact the phase where we have the bursting out. The other phase, lysogeny, right? So we're incorporating the gene into the genome. Or what is the term for viruses that can cause cancer? All right, oncovirus is correct. Good job. Good job. All right, what kind of genome do prions have? So I mentioned about the different kinds of germs you can have. Some of them don't have cells. This is one of them, a prion. So what kind of genome do they have?
Did I black out during this class? Like, oh, it was fake.
Yeah, it was. Yeah, they don't have a genome. So yes, they are just misfolded proteins. Exactly. They don't have any kind of genome. They're literally just like.
God, that picture.
Okay, so this is our halfway point. That was all of unit 1. Unit 2 is going to cover, this is the heavy one. Yeah, now we're moving, that was the intro stuff. Now we're moving into the genetics and the metabolism chapter.
Cute fact.
Unit 2 is, I know, right? Unit 2 is going to have a lot of info. Genetics, genetic engineering, microbial nutrition and ecology, metabolism, disease and epidemiology stuff, right? before we move on, I would like to kindly inform all of you that you guys have a 6% higher grade average than all of my other sections.
Yeah.
You wanna guess why? It's because you show up and you try and you do your work.
I love
yeah.
you.
I think it shows, honestly, like, as soon as I look at the average, I'm, man, why are they so much higher? I'm like, oh, wait, no, obviously they would be higher because they try a lot harder than the other classes do. I'm not saying that the other folks just overall just don't try at all, but there are enough students in those sections that aren't, that it's dragging down the average. I'll say that for my online course, it's the worst that I've ever had an online course ever perform, ever, by far. Just because they're just not self-motivating this semester. And what I find is that when you have kind of a core group of people that get along well at studying and put in the effort, that it carries for the rest of the section. Like the rest of the section sort of like mimics that. In that class, they have a core group of people that are just outwardly and proud of it lazy. So it's carrying through that section, but this is clearly the example of the first case scenario where I feel like, you have people who are working hard, trying to do the best they can, and it's carrying over to other people. So I just want to commend you guys for that. Yeah, I think that it does show the hard work that's putting in.
I will say you are a professor worth showing up for because you actually care about us and it shows. And like, it's been really, really good semester.
I do really appreciate
We're
that.
actually
That's
fun.
what I feel like about you, like you just got through the bullshit, like actually real down to the earth, like.
Yes.
What?
Please
You guys don't
stop
need
me.
to know this shit next.
Of course, and the way that I look at it is I know that the class is hard, and of course, like my goal genuinely is to move towards making it easier, but not giving up like the
Don't make it
learning
easier.
part of it. Yeah,
It's fun.
if I can somehow maintain the learning, and I think it's not about like what so much like we do, what so much we do in the class as much as it is about like teaching, like learning styles of people, like people coming to the class and what they need, whether they're visual learners or engaging learners or instead of hands-on type people or whatever. I can't always accommodate those types of people in all the classes. So I think that has part of it, but I'm trying to learn what I can do to make it easier. And believe me, I'm working on that always. But it's people who succeed that help me just as much as people that don't. If not more so, because at least like you're working and you're learning and then you have an idea of like what you would need more of, and that helps me learn. So I thank you guys.
Everything you succeed. If you're doing extra study sessions with us when most professors wouldn't.
Yeah.
I don't know any professor that comes to each chapter for review.
Or
Or
shoot,
anything
a
like
review
that.
in general.
Or shows up.
Or so talks about the content instead of
Yeah,
course
actually
code, yeah.
talks about it, actually tells you what you're supposed to be studying.
Yeah.
Correct my homework.
Yeah.
Literally respond on Messenger, like, if you're a really,
I
really good
try.
professor.
I really appreciate it, you guys. I really do. Because I mean, I know you know this, but I care. I genuinely care. And that matters a lot to me.
It
So
shows.
it's been really wonderful having a class like you guys, just because they don't come all the time, and not all my sections are like this. So I actually look forward to this. I'm like, yeah, okay, the Monday morning class, and I'm glad that I have you guys first before the others. Yeah.
Before everyone else would.
And I have you guys usually the lab, yeah, I have you guys' lab right before the online people. So it's like the good that I get with like interaction with you guys in lab or whatever, I have to like have that carry me over through that lab. Even though I like them as people. They're great, they're fun, but like, I'm just like, I can't just joke with you and you get A's. Like that's not.
How's your crappy student doing? Thank you one.
She's smart. That's the problem with her, is that like she can. You know what I mean, though, like she's one of those people who cheated on the test, but like probably didn't need to. Like if she just studies, she does really well. But she just thought she knew better. And since I couldn't prove it, I have no actual evidence other than like some student basically confirming that, yeah, I'm pretty sure she had her phone out, but I don't have proof. I didn't catch it. So all I can do is just put her in the position where she can't do that. And now she knows. So I'm pretty sure that like
She's studying.
she'll study. Yeah, she'll have to study.
Is
But
she
maybe that's a
doing
good
well?
thing too. She has a B.
Okay.
So yeah. It's just in the lab stuff. She doesn't do well in the lab, really.
I also don't do well.
Yeah.
Yeah.
That's fair.
Yeah. Anyways. Yeah. All right, let's get through this second half of this, and then I will let you guys go. All right, Jenny has brown hair, but she has one brown gene from mom and one blonde gene from dad. Her brown hair color is her what? She's presenting with the brown hair, even though she has one brown and one blonde gene. So what would that make for brown hair? All right, excellent. I'm so proud of you guys for that one. That is phenotype. So it's what's expressed, right? All right. Which one term describes DNA replication best? If you had to pick one of them.
That's good.
DNA replication.
Oh.
Oh.
Now, of course, if you guys are taking this on the test, you'd have a little bit more time to think through it. But the answer is semi-conservative. The reason it's not transcription is because transcription is DNA to RNA, right? Because we're going from DNA to RNA, we're making something else entirely different. That's transcription. Non-canonical, obviously glad that nobody picked that one. Nuclear, even if we were saying nuclear, like bacteria don't do DNA replication in a nucleus because they don't have a nucleus. So we couldn't even apply it being in a nucleus, even if that was made clear, to DNA replication because it doesn't always happen there. So semi-conservative, right? We have one old strand and one new strand every time that we make a copy. So we know that happens no matter what. All right. True or false? The building blocks of DNA and RNA are known as nucleotides and consist of a phosphate, a pentose, and a nitrogenous base. True or false? This horrifying picture.
Looks like dinosaur.
Oh, it's one of them bugs that you poop.
Yeah.
The stranger, y'all know what I'm yapping about.
It's supposed to be, I think, like transcription or something like that. Anyway, so this one is true. DNA and RNA are both made of nucleotides. That's just their building block. That a nucleotide is a phosphate, a pentose, and that's either going to be deoxyribose or ribose, and then a nitrogenous base of some kind, the ATCG or the AUCG.
Yeah.
All right, true or false? Exons are non-coding regions in eukaryotic DNA that must be excised to make a usable mRNA transcript.
I think I bet it's 25% chance of that. Almost true.
All right, so I like that the trend was to false, which is the correct answer. And it says exons are non-coding regions. That's not true. That's the false part, right? Introns are. And we splice the introns out to join the exons to make the reading frames. Cool. All right, what are codons used for? All right, so good work. This could have been worse. So they are the 3 nucleotide code that tells which amino acid to attach next. Now the next popular choice was the green one. They are the 3 nucleotide sequence on DNA that corresponds to an amino acid, but that has its own terminology. That's a triplet. So on DNA, it's a triplet. On messenger RNA, where we're actually reading it to make our product, that's going to be the codon. So well, that's why we're doing this, right? To help you parse out those little details. Okay, the codon ACA codes for threonine. A mutation change becomes AGA, and now it's arginine. What type of mutation is this? All right. So anytime we change an amino acid because of a mutation, that's missense. If we are changing an amino acid into a stop codon, that's nonsense. So we're not making any sense at all at that point. There's no amino acid at all being made. Silent means we're keeping the same amino acid even though the nucleotide sequence changes. And then obviously fun sense is not a word. Sorry. We have to ruin all the fun. All right, what types of DNA sequences do restriction endonucleases tend to cut at?
Start or stop.
That's the start. Not that I'm going to test you over it, but yeah. All right, so this palindrome, good job. Same forward as backwards, yeah. It's the same forward on the top strand as it is backwards on the other strand. All right, what is the purpose of PCR? What does it do?
Why it takes 45 minutes for us to do it.
All right, so the whole point of PCR, what it does, it's replicating a specific known DNA sequence. You have to know what you're targeting in order to run this. So no antibiotic susceptibility, no antibodies are involved, and no proteins here. This is all DNA replication. Just making copies of DNA. All right, the second step of PCR is anneal. What is being annealed? Denature, anneal, elongate. What is anneal? So anneal means to stick something together, by the way. So this one's the primers that are annealing. I actually showed it in the previous slide, but I just didn't want to give it away too easily on this one. The primers that are targeting the specific gene that you're interested in are being annealed on there by lowering of the temperature. And then you have to have something to build off of to add your nucleotides to. So that's why we need the primers in the first place. In DNA gel electrophoresis, what is causing the DNA fragments to separate by size? So we know they're separating by size. In fact, you may even remember that the small ones run faster. But what is causing them to separate?
Oh, yeah.
All right, so this is the flow of electric current. The electric current is what's moving the DNA fragments to the positive end. Remember, they move to the positive because they themselves are negatively charged. That's from phosphates. All right, BT corn produces proteins from bacteria toxic to certain insects, but not harmful to humans. What does that make it? BT corn. Could be more than one.
Reminds me of that one picture, or the one video of the one girl eating corn on a grill and it's catching in her hair.
Oh, yeah. Yeah, I forgot about that. All right, so this one is genetically modified organism and transgenic. They're the same thing. So we've put BT, by the way, BT corn has genes inserted into it from bacteria, Bacillus thuringiensis. And that's what gives it that ability. Pretty interesting. All right, true or false, somatic gene therapy can be passed down to future generations. All right, so this one is false. Does anybody remember the term?
No.
Yeah. Okay, the term for gene therapy that can be passed on, it's called germline. Okay,
Oh.
that makes sense. Yes, of course, when we say it
Uh-huh.
out loud, but yeah. What is the term for an organism that gets its carbon from inorganic sources and its energy from the sun? Yeah, I don't care that much about these. I try to put fair questions. Alrighty. So this is photo, autotroph. Photo is sun, energy, and auto means they're getting the carbon, inorganic carbon, and turning it into organic. So doing that themselves automatically. So what is the term for the nutrient that must be provided in diet because an organism can't make it for itself? Remember the term for that.
None of these are what I thought of.
All right, good job. That's gonna be growth factor. Anything that organism needs that it can't make for itself. What type of solution could have a typical cell swell up and burst, just like this balloon if you blew it up? Putting a typical cell in what kind of solution and it'll burst. All right, so at least we're on the same page that it would have to be hyper or hypo. The answer is hypo. And I'm just gonna draw a little picture really quick about it so that we are all understanding about that. This is a cell. It's got some crap in it, right? Whatever. And here's also a cell that has some crap in it. All right. In hyper, we have a whole lot of crap on the outside, right? Just a ton more concentrated, like when you pack something in salt, right? So the water that's in here, which you can't see, because I can't draw, because that's not the crap we're talking about, that's the only thing that can cross the membrane. The crap can't. So it wants it to be equal concentrations on both sides. So it will get more concentrated in here by letting out water and watering down the outside. So this will shrink, right? This, however, if there's like pretty much next to nothing out here, right, it wants this to be more concentrated and this to get watered down so they match. So it'll keep taking in water from out here, basically, until it'll swell and swell and swell and swell with water to try to dilute out. And it can potentially keep doing that until it bursts the cell. So that is the hypo. Hypo, and it's hypo because that means lower, right? There's less salt outside. Hyper is higher because we have more outside. Make sense?
Yep.
Well, at least for now.
Yeah, for now, don't forget about it.
Right.
Until you ask. Yeah.
All right, pathogens and microbes of our biome thrive at temperatures near around room temperature and body temperature. What does that make them? What term? All right, it's gonna be mesophiles. That's meso being middle. What role do black bacteria play in the nitrogen cycle? All right, so they're gonna convert it to a form that plants can use. That's great. Great job. What are coliforms?
Well, I brought it for change.
Most of you guys got this one. This is going to be bacteria that if they're there, the pathogens could potentially be there too. Even if you test and you don't find them, they could later on if they're making it through. So what is the term that refers to chemical reactions in the body involving breaking down molecules into smaller products? Breaking bonds. All right, so this one's catabolism. The cats tear things apart and break things apart, just like your metabolism. Yeah, Enzyme-
Pictures on our best.
I know, right? Enzymes help overcome activation energy of starter product. What is the correct term for the starter? So if we're acting on a starter to turn it into products, what is that we're acting on? This is an actual like animation of what it looks like when proteins carry vesicles full of stuff.
Yeah.
So this is substrate. That's correct. That's the word for this. I don't know what else to say about it. A term. All right, what does ATP stand for? And this is ATP synthase, by the way, which is pretty cool to see it operating. Pretty much, I feel like two that you guys are gonna get stuck between. Yeah, I had a feeling. So adenine triphosphate and then adenosine triphosphate is just referring to whether there's a sugar bonded in there. So the sugar is going to be there. That scene is what indicates that. So it's just a matter of remembering that term. I'm not going to put them that closely worded on the test, by the way. So yeah.
Thank you.
Yeah. All right, in aerobic respiration, what are the 3 stages of energy production? Pick the 3.
Oh my God.
Alrighty, so most of you guys got this, did really good on it, actually. Electron transport chain, Krebs cycle, and glycolysis, of course, starting in the reverse of those. Right. What is the final electron acceptor in aerobic respiration? Final electron acceptor.
This music is not doing me any help. He does it. So rude.
All right, it's gonna be oxygen. Good job, guys. Nice work. All right, true or false? Fermentation generates just as much energy per glucose on average as does aerobic respiration.
Oh, I hate it.
Your views are on point.
All right. All right, so this one is false. Fermentation only goes through glycolysis, and that is it. And glycolysis only makes 2 ATP. Not that I expect you guys necessarily to remember that right now, since I know that we're just started studying, but yeah, it only makes 2 ATP. Does anybody remember how much ATP you can make with aerobic respiration overall?
Isn't it 2 and 2?
So you will have 2 made in glycolysis. You can have 2 for each round of Krebs. But then once you finally get through the electron transport chain, 38 is the total. So it's quite a lot. Yeah. So fermentation is next to nothing. But if you have oxygen available, you'll go through aerobic respiration.
No.
Based on what we've learned, which organism would be a true pathogen? A true pathogen.
Oh, I hate this word.
I need your bird here.
All righty, wonderful. So it would be the plague. One thing that we notice about plague versus all of these is that it always and only ever causes disease, right? It's not ever going to be part of your normal biome and be what we might consider a protective thing. Candida albicans yeast can be that. So can Staph aureus, and so can Streptococcus pneumoniae. That can be part of your biome, and then therefore be some of the bugs that are sitting in the stadium seats that are helping protect you from bad guys. So why pestis is always a bad guy, is always a pathogen. So that's what makes him a true pathogen. Not ever going to be opportunistic. Make any healthy person sick. All right, define virulence factor. All righty, good job. So this is something that increases pathogenicity and infectiousness for whatever that pathogen is, right? So our other choice that was popular, something that makes viruses better able to infect cells. That's true also, right? It could be if you have something for a virus that helps it infect cells better, that could be considered a virulence factor, but it's not the definition of virulence factor, right? So that's the trick there. That's what is catching us up there. All right. Which term refers to an agent which came from outside of the cell or the body? This is a pretty. All right, exogenous, excellent. Glad that we're learning. It's something. All right. This could be more than one choice. So which of the following are symptoms? We knew this crap was coming back. This is my favorite, by the way, this one, this meme. And if anybody knows where this is from, I applaud you. All right, so the symptoms here, some are signs you can actually measure and are tangible. Symptoms are things that can't. So headache and fatigue, these are things you have to just trust the patient. And for what it's worth, that is Saturday Night Live. And it was when Will Ferrell used to be on Saturday Night Live and Christopher Walken was like directing the band or whatever. More cowbell, yeah. That's where that came from. Huh?
You know.
Lymphadenopathy, that is swollen lymph nodes. So yeah. And if you get to a term like that and you're like, man, I feel like I should know this, but I don't know what it is, and you can ask me if I feel like you should know it as part of terminology, then I'll just tell you no, but that's one that I would tell you because that's not what I expected really like quiz you on or something like that. So if you didn't know it, or there's a term on there, other medical wise that you guys haven't learned, you can ask. It doesn't hurt. Okay. So it doesn't occur to me sometimes. All right. In which phase of infection are the very first signs and symptoms seen? All right. Yeah. So we've got all four phases up here as possible choices. We've got the incubation. That's where we at first got introduced to whatever the pathogen is, and it's just getting, set up and growing. We haven't reacted to it yet. The prodrome or the prodromal phase, this is where we start having the first symptoms. Acute or infection phase, this is going to be the height of the symptoms. You're going to have the most of the organism growing in your body during the infection. And then convalescence is whenever we have dying off of the symptoms as the organism is cleared from the body. So that's what those phases are. All right, a fly flies out of a porta potty and lands on your food. It's not infected, but you might get sick. So what is the fly in this scenario? Right, for sure, that, yeah. Now, please tell me you guys know this one.
Every time I see my I think of just a little weird. Oh, I got it right.
All right, so this one's the mechanical vector. So The reason it's, so etiological agent is the thing that is actually causing the disease, I don't even know what, salmonella or whatever it could be, the E. coli, that would be the thing actually causing the disease. Fomite, those are gonna be non-living objects that can be used to transmit things as non-living vectors. Then we have biological vector versus mechanical. It's mechanical because it's not infected, right? So it's just carrying it on its little feet. Like whenever you go take your dog out in the yard and you chase after it because it won't come inside and then you step in its poop and then you walk inside and you didn't realize you had poop on your feet. That's you being a mechanical vector.
That's why you don't wear shoes in the house. No papa shoes.
I have shoes specifically for going in my backyard because of that. Who was involved in identifying the source of the cholera infection in 1800s London and containing it? It's like one of the only scientists I would, yeah. All right, so this one,
I could tell from your name and also it was day or two.
yeah, it's John Snow, different John Snow, obviously, but that's his name. So, Alexander Fleming did penicillin. Joseph Lister, he cleaned the, surgical suites with, yes, Listerine man. And he used, do you guys remember what he used to do that? Phenol or carbolic acid, right? And then Florence Nightingale, anybody know what she did? Also epidemiology, she was a nurse, yeah, and she figured out that basically a lot of people were getting sick in the hospital from being touched in the hospital by people not washing their hands. So she helped alleviate that. Wash your freaking hands.
I think it's so gross, you have to remind people to do that.
I know. All right. Which term refers to the occurrence of a disease in a given time frame? So people, how many people get infected in a month or something? This is a multi-select, so there could be more than one that fall within this, if you so choose. All right. So this one is morbidity and incidence. So prevalence it is related, right? Prevalence is dealing with the number of total infections for that outbreak, basically. So as people are getting, sick overall with that outbreak. And morbidity and incidence both are referring to how many people are getting sick like per month or presenting with symptoms in that time frame. They're kind of the same thing. I was like, why isn't it doing anything? Sorry, no offense, but he didn't get it right. So that's why. All right, a cholera outbreak in the 1800s, where people got sick from a water pump over several weeks, is an example of what kind of outbreak? Stuff that we haven't thought about in a little while. Righty. So this one is common source epidemic. Point source is like if somebody had come in here and just like, I don't know, started coughing and coughing and coughing, and then we all got COVID or something from them, right? All of us got sick at that one time, that one place, all at once. Whenever we have common source epidemic, it is one source that we're talking about, which is this water pump. But people are visiting it over weeks, but it's still the place where they're all getting sick. So since it's spread out over time, it's common source, but it didn't happen at a single point in
So
time.
point sources, like one person, one time infects everyone.
Or like if there was like an infected water pump that like everybody went to it to go get water at that moment, but somebody instantly realized it was fixed or icky or whatever and shut it off right away, and everybody got sick from that one encounter, that could fall into point source. It'd be weird, but like, yeah.
So it's like
One, yeah, or exactly.
if that one person like went somewhere else and coughed, that would be common source because they're going with different people, or?
That's a good question. Potentially, yeah, I guess depending on how you would break it up. If they went, I feel like if they went enough places.
Yeah.
So if it was just like maybe one or two, you could still potentially count it as a point source. It just depends on how the epidemiologist would view it, and how related those incidences were. But yeah, especially if it's spread over time, like a greater amount of time. So if we're talking about one person who had a new version of COVID that they got visiting China, and they come back to the United States and they're like, go to class, and they go to all their classes, and then they go to Walmart, and then they do this, and then they're still sick, and the next day they go do more of the same stuff and whatever, and they continually keep spreading it, then that could be a good example of that, yes. All right. Good question. An outbreak that exceeds the expected prevalence, but which doesn't cross continents is known as. So having a greater outbreak than what you normally would see in that area. Maybe you usually see none. But it's not crossing continents. All right, so this word for this one is epidemic. It's just a matter of being familiar with those terms. And so endemic is something that normally is there and it's at the level that you expect it to be at. Epidemic, now we've exceeded that expectation of what normally is there, or we have something completely new. So obviously then that would be outside of what you would expect it to be. Sporadic is just you have 1 or 2 cases. Pandemic, you have to cross continents. All right, 74. What does it mean for screening or diagnostic tests to be highly sensitive? Could be more than one choice. In fact, I'll say that it's 2. All righty, so false negatives are less likely because we want to pick up every single positive that there is, even really weak positives. We want to be sure that we catch them. So that's really, really sensitive, right? Also, the test will trend toward identifying any possible positive in a given sample. It's another way of wording it. So avoiding the false negatives, we're not calling something negative when it really isn't, or, you know, trending towards identifying all of the possible positives. It's kind of the same thing whenever you break them down. That makes sense? The other one, you know what it applies to, right? If we look at it, that other side of it, that's going to be more towards not the sensitivity, but the specificity. I know those terms are confusing sometimes. What is the term for how many people in an infected individual is likely to infect in the course of their illness? So if you're sick, you've got COVID, and you're likely to infect, according to the math via epidemiology, 5 people.
Like an artwork, but I don't know.
Yeah,
And doctor's office.
I kind of like it. So R naught is the term for that, or R with the little 0, right? R sub 0, same thing. All right, so that is units 1 and 2. We are done. Do you guys have any questions about any of this? Anything else coming up? Because I know we still got 3 and 4 on Wednesday. No questions? Cool. So I will post the recording and the link to this Kahoot once the recording is available. So,
Thank you.
yeah.
Thank you.
You're welcome, you guys.
If you guys are interested, a couple of us are gonna get pizza after class on Wednesday, is it Wednesday? Monday. What's the last day for our exam?
Monday, yeah.
We're gonna- Monday, we're gonna do pizza if you guys would like to partake in that.
I'm bringing cupcakes.
Yes.